The following presentation was prepared by Shama, Sarah S., Sarah U., Ben, and Nicole. In this video, we will be discussing various aspects of a rare group of eye diseases called retinitis pigmentosa, such as modes of inheritance, frequency measures, major phenotypes, testing, and treatment. Thank you in advance for listening. Retinitis pigmentosa describes a group of genetic disorders that damage the light-sensitive cells or retinal pigment epithelium in the retina, leading to gradual vision loss over time as the cells begin to die. The light-sensitive cells are also called photoreceptors. They absorb light and translate it into signals. The light absorption contributes to the photoreceptor signal output that is interpreted by the brain. The photoreceptor cells include rod cells and cone cells, responsible for peripheral and night vision, and high acuity vision, detail, and color, respectively. This disease is also known as rod-cone dystrophy. There are many ways that non-syndromic or simple RP can be inherited. Non-syndromic means that the disorder is not associated with signs and symptoms that affect other organs or tissues. It's often inherited as an autosomal dominant disorder, meaning that males and females are equally likely to inherit the altered gene, and that inheriting a single copy of the altered gene is enough to cause a certain genetic condition. Retinitis pigmentosa can also be inherited as an autosomal, recessive, and X-linked disorder. However, these forms are not as common. Also, the disorder is very heterogeneous, making it difficult to correlate genotype with phenotype. An alteration in the Rho gene is most commonly associated with autosomal dominant retinitis pigmentosa. The Rho gene encodes the rhodopsin protein, which is the most abundant protein found in the rod cells. Rhodopsin is a G protein-coupled receptor, meaning that when light activates rhodopsin, a series of events take place in the visual transduction pathway, ultimately stimulating biological processes in the nervous system, thereby allowing an organism to sense light. More than 100 different variants of the Rho gene have been identified. However, a common alteration identified in approximately 10% of Americans with autosomal dominant retinitis pigmentosa is a substitution of proline to histidine at the 23rd amino acid position. So far, more than 70 different genes have been identified that can lead to non-syndromic retinitis pigmentosa, with over 3,000 different mutations.
We will now talk about frequency. Retinitis pigmentosa is the most common inherited disease of the retina. It is estimated to affect 1 in 4,000 people in the US and 1 in 5,000 people worldwide. A retrospective study in Korea from 2011 to 2014 showed people with retinitis pigmentosa have a higher mortality risk compared to the general population. Retinitis pigmentosa individuals had a standardized mortality ratio of 1.56 for all ages.
So as we said before, retinitis pigmentosa, or RP, is progressive degeneration of the retina from mid-periphery to central, and it can sometimes involve the macula and fovea, and what you'll find is bone spicule formations, attenuated blood vessels, reduced visual field, abnormal electroretinograms, changes in OCT, and changes in visual function that are very individual to the patient. And so I have two photos. There's one of a healthy retina and a retina affected by RP. And as you can see in the one affected by RP, there's those dark brown spots, and so those are the bone spicule formations. And if you compare the arterioles in the healthy retina as compared to those in the one affected by RP, they're far thinner. So in this slide, I really wanted to show the disease progression of RP. And so you can see in the early stage that there isn't too much that's very apparent, and it looks somewhat like a healthy retina. And then you see mid-stage, and you really begin to see the pigmentation come in, and you can see the arterial narrowing. And in the late stage, the bone spicules, the super dark pigmentation is very, very apparent, and you can barely see any of the arterioles. So I know that we've talked a lot about non-syndromic RP, which is confined to the eye, but there's also syndromic RP, and it has non-ocular symptoms, and so I've left the most common ones there, and then some rare syndromes with clinical importance, and that means that if you find them and you treat them early enough, you can save a patient's vision, and I thought that it was pretty cool, so I've left it for your interest. So RP can be highly variable, but the general progression of the disease begins with night blindness in adolescence, and then loss of mid-peripheral vision in young adulthood, and then loss of peripheral vision, also called tunnel vision, and then loss of central vision at around 60 years old. Most patients are considered legally blind around 40 years old, and while there is increased mortality in RP patients, it's due to the functional impact of the disease and not the pathological disease process.
We will now go over a few ways retinitis pigmentosa is diagnosed. First, we will discuss a simple eye exam, which is the dilated eye exam. Here, the doctor uses eye drops to dilate the pupils to see the back of the retina more clearly, letting more light in. Then the doctor checks the patient's peripheral vision with a visual field test to observe if the patient can see things on the side without moving their eyes. Electroretinography. Here, a recording electrode is placed on the cornea using a contact lens or on the lower eyelids. Then the eyes are numbed and conductive solution is used to protect the corneal surface. Next, the electrical activity of the retina's response to the light is measured, and in retinitis pigmentosa, responses are decreased. Next, we have optical coherence tomography. With this method, light waves take detailed pictures of the retina. There's the picture on the top, which shows a healthy retina where all the layers are present, and on the bottom, it shows a retina that is deteriorated. In fundus autofluorescence imaging, intrinsic fluorophores are seen in the retina, which is made of lipofuscin. Lipofuscin is located in the retinal pigment epithelium and accumulates when the retina is broken down and is a result of aging. The healthy retina, shown on the left, has no granular spots or increase in fluorescence, and on the right is a picture of retinitis pigmentosa retina, which shows granular spots, increased fluorescence in the nasal retina beside the optic disc, which is also known as nasal sparing and is shown by those arrows and is specific to retinitis pigmentosa. And there is an increased fluorescence around the fovea, which is the brighter circle on the left. Lastly, there is genetic testing. The doctor may suggest genetic testing to learn about what type of retinitis pigmentosa the patient has and can tell how their symptoms may progress or change over time. The method that is used is PCR and bidirectional sequencing with Sanger sequence analysis.
Now onto the therapy and management of symptoms for retinitis pigmentosa. Currently, there is no cure for this disease, but there are ways to help manage and slow the progression of it. Supplements like vitamin A, fish oil, and lutein may help slow vision loss. Use of UV glasses can help delay the start of the symptoms of retinitis pigmentosa. And for patients that are in the late stage of this disease, retinal implants is an option for them. So these retinal implants are microchips that are placed on the retina and is paired with special glasses that can convert light detected by the implant into signals that are sent to the brain for processing. This implant is called the Argus 2, and its setup and everything is shown in the bottom right image. There are more treatment options, but they are currently on clinical trials. One of these treatment options that are in clinical trials is gene therapy for the RHO gene. The RHO gene codes for a protein called rhodopsin, which is vital to phototransduction. A mutation in this gene causes a faulty version of the rhodopsin protein to be produced, and there are over 120 different rhodopsin mutations that can cause this faulty protein to be made. In addition, this protein is toxic to the retina, which causes degeneration of photoreceptors, which leads to retinitis pigmentosa. A new treatment is being developed that uses targeted ribonucleic acid to silence the mutant allele, which in turn allows expression of the wild-type allele, and it has been shown to be effective in preserving retinal activity in rats. This method of treatment is achieved by using ribozymes that have specific targets for mRNA. Because there are over 120 different mutations to this protein, an alternative method was designed to degrade all rhodopsin mRNAs and replace them with a healthy RHO copy. This universal method uses small interfering RNA, which works by degrading mRNA by being initially introduced as double-stranded RNA, and then being cut to become small interfering RNA. Then, it is incorporated into an RNA-induced silencing complex, and from then the complex binds to complementary sequence on the target mRNA and induces its degradation, thus silencing it. The replacement gene is then delivered by an adeno-associated virus as a vector. The methods above are used to treat RHO-linked RP.